This protocol facilitates investigation of the mechanisms underlying the development and disease of axon bundles using motor nerve organoid derived from the human iPS cells outside of the bodies.
By simply culturing neuronal spheroids in custom-made microculture chips, unidirectional axon bundles can be spontaneously generated and be used for various downstream experiments.
Motor nerve organoid can facilitate the screening and testing of the drugs for motor neuron diseases, including ALS, by providing a more physiological model than a previous in vitro systems.
In a fume hood and wearing the appropriate PPE, dispense 3 milliliters of SU8 2100 onto an acetone-cleaned silicon wafer and place the wafer in the center of a spin coater. Fix the wafer by vacuum and spin the wafer at 500 revolutions per minute for 10 seconds to coat the SU8 uniformly across the wafer surface. Then spin the wafer at 1,500 revolutions per minute for 30 seconds with an acceleration of 300 revolutions per second to obtain a 150 micron thick layer of photoresist on the wafer. After soft baking, set the photomask to the mask aligner and expose the wafer to ultraviolet light for 60 seconds. After baking on a hot plate, develop the wafer for 10 to 20 minutes in photoresist developer with agitation on an orbital shaker, changing the developing solution once during the process.